Yes. So I just started looking at the top 10. Yes. Okay, that's fair. More if there was somebody else. Yeah, we were we got Gabe out here. Gabe,
Great. No, so I've actually worked on
Yeah.
Yeah. No,
That's what you're here for.
Yes.
Oh,
But we don't have whenever you're ready.
Yeah.
Oh, do you want to finish it?
No. I just want to say something.
Okay.
Well, I don't mind waiting on someone. I'm just relaxed.
Yeah, we'll just keep. Okay, guys, so I'm going to get started now that we're already 10 minutes into the meeting. I'm very excited to announce our speaker today, Dr. Gabe Rocklin. He earned his bachelor's degree in biology, chemistry, and history from Claremont McKenna College. And then after that, he went on and did his PhD from UC San Francisco with Dr. Shoykin and Dr. Bill. And most recently, he did his postdoc with Dr. Baker from UW, where he did a major protein design project. And he is the newest faculty hire at Center for Synthetic Biology, and I'm very happy to have you here.
All right, thank you so much, and thanks for coming and for the invitation. Can everyone hear me? Because I'm not using the microphone. Okay, great. So this talk is going to be a little bit of synthetic biology, but it's not mainly a synthetic biology talk. I'm going to talk a lot about cool, high throughput biophysics that we're doing, and this is a new project where I'm really excited to recruit someone to work on this project. So if you're a first-year student who hasn't picked a lab yet, come on to a summer rotation, or if you have a friend who's going to start in the fall and they're looking for a cool lab to rotate in, then this is, I think, going to be a great project that that someone can take the lead on. So there's going to be basically 3 parts to the talk. At the very beginning, I'm just going to give a quick introduction, quick to how pointers work. Quick introduction to de novo protein design, because maybe you weren't here a few weeks ago when David Baker gave his seminar, but also we're using de novo protein design to answer questions about protein biophysics. So I'm going to review some old published work just to give the context for the rest of the talk. Then, so I'm going to introduce de novo protein design. Then I'm going to talk about the old work on developing a high throughput method to screen de novo proteins for folding stability. But then the bulk of the talk is going to be the new high throughput approach, not just to measuring folding stability for proteins, but for measuring their energy landscapes. And I'll tell you in more detail what I mean by that when we get there. So as a quick introduction to de novo protein design is the creation of entirely new protein folds without using natural starting points. So what that means is that we'll start with a kind of protein diagram that specifies what the structural elements of our final design are going to be. Like there's going to be 3 beta strands, and the first strand is 6 residues long, and then there's a loop, and then there's a helix, and so on. And we turn this diagram into a full 3D protein. And the goal of developing technology to do this is that we think in the long term that if we can design the entire protein structure, then we'll be able to design more sophisticated functions than if we're stuck just modifying the structures that nature has given us. And so de novo protein design basically has 3 stages. We start with this blueprint, and now I've written in actually the lengths of all these elements. So this helix is 14 residues long, and so on. And then from this blueprint, we construct a 3-dimensional backbone. So this is not a protein, but it's a 3-dimensional version that 3-dimensional version of this basically, where now, rather than just being a diagram, all the residues are positioned in space. And we use the Rosetta software to do this. And then once we have the 3-dimensional version of our diagram, then the last step is to sample many possible protein sequences. Sample all the possible side chains at all these different positions in space to find a protein sequence that's going to be very low energy in that folded structure. So then that's the sequence that we would experimentally express and test. Now, a couple of years ago, I started a high throughput protein design effort where basically I took advantage of really cheap DNA synthesis. So if you want to make a protein, if you want to design a protein on the computer and experimentally test it, you need to synthesize the gene for that protein. And now we can synthesize tens of thousands of genes as long as those genes are fairly short. So I'm basically talking about long oligos. So I designed thousands of proteins in each of these 4 different overall shapes. And each protein was small enough that it could be encoded on a single oligo. And I ordered those oligos 10,000 at a time, and then screened all those proteins for folding using a protease resistance-based assay. So not all the designs that come off the computer are good, but we could test each design for whether it folded into a stable structure, because if it folds, it gets digested by protease more slowly than if it's an unfolded structure, then it can get cut up by protease very quickly. So we designed thousands of things in each of these shapes, and we found a few thousand that were stable. And I just want to emphasize one more thing about de novo protein design. When we design different proteins that have this shape, their sequences can actually be quite diverse. So if you take 2 of the proteins in the set that were both designed to fold like this, their sequence identity is maybe 50%. And some of them will be much more diverse than that. So think of them as protein families. Like if you look at homologs of a protein that fold the same way, you'll often see sequence identities in the 20 to 30% range. And that's kind of what we see here. So we're not designing like just a few point mutants that make the difference between one sequence and another. These are very different sequences, but they still fold into these same overall structures. And the cool thing about designing these thousands at a time is that we could really systematically examine the differences between stable proteins and unstable proteins. And what that let us do was to try to improve our design method over time. So the first time that I tried designing any of these proteins, this is round 1 up here, there was about a 20% success rate in one of the categories, and then almost no successes in the other categories. But we examined these successes and what made them different from the failures. We saw we needed to, for example, make our proteins more hydrophobic and a few other things. And then the second time we did this, well, you can see the second time we did this, we got a much higher success rate for this protein fold. And what we learned on this fold was also transferable to getting successes in one of the other different folds. And then over the course of 4 rounds, we eventually became really successful in 3 out of these 4 overall folds, getting a couple thousand stable designs. And Tayan, my grad student right there, is currently figuring out what's going wrong with this fold, and he's trying to design better proteins in this fold. That's just a small project. Okay, so we learned from our successful designs, we made better designs over time. One of the other cool things that we could do with these designs was redesign the surfaces after we know that they're stable, redesign the surfaces to bind to different therapeutic targets. So this is a very small protein where we redesigned this surface so that it would bind to influenza hemagglutinin. And if you inject or if you spray this protein into mouse nostrils and then give them a flu challenge, then they don't come down with the flu because this protein is binding hemagglutinin and deactivating it. This is another one of these mini proteins that we redesigned to bind to botulism hemagglutinin. And and again, it binds and prevents an association with a target botulism neurotoxin. It prevents that molecule from carrying out its function. So this is one of the near-term applications of these mini proteins is for this type of therapeutic design. One of the other cool things that's come out of this data set is a lot of other groups that have no connection to Rosetta or the type of protein modeling that I do are starting to use this high throughput data set to optimize their own protein design methods, especially protein design methods that are using deep neural networks. So just in the past, like 6 months, there's been a couple of preprints from really nice groups. This is the Gregorian lab at Dartmouth, George Church's lab at Harvard. This isn't even a preprint yet, but you can read their manuscript on GitHub. And all these are taking advantage of the first really large-scale data set on successful and failed protein designs, and they're using it to improve their own methods. And I think that's the way that I like to do science, is to produce large data sets that are going to be useful for everyone, try to learn as much as we can, but also share as much of that data as we can with everyone else so people can go in different directions with it. And that's really, I think, taken off with the protease work. Okay, so for the rest of the talk, I'm going to tell you not so much about design, but about high throughput determination of protein energy landscapes. So as you all know, proteins are not just in one folded structure or one unfolded structure. They're always fluctuating between many different conformations, and those conformations can have different energies. So some of those conformations are populated very frequently, some are somewhat rare, and some are extremely rare. And each protein is always moving between these different conformations. And what those conformations are and what their different energies are is what I'm calling a protein energy landscape. And just to give an example of this importance, in biology and medicine, there's a movie on the next slide, so it loads slowly. All right, this is the protein human lysozyme, and there's two different versions of this molecule shown here. So in white is wild type human lysozyme, and in red is a mutant that's an isoleucine to threonine mutant that happens right here. And you can see from the crystal structures of these two proteins that they're completely identical. So the mutation doesn't change the crystal structure at all. But for a very small number of people that have this mutation, it causes human lysozyme to form amyloid fibrils, and it basically destroys your kidney, your liver, and it's a really bad disease that's fortunately pretty rare. But you can see that it doesn't change the structure of the protein at all. And not only does it not change the structure, it doesn't change the function of the protein either. But it really all has to do with how it changes the populations and the different conformational states that this molecule accesses. So you might think, well, maybe the mutation is destabilizing, so it causes the protein to unfold a lot. And actually, if you look at the distribution of folding stabilities of soluble proteins in the human proteome, 0 means you're pretty unstable. These are extremely, extremely stable proteins. Lysozyme's out here normally. But even with the mutant, even the mutated destabilized form is still one of the most stable proteins in the proteome. Like, this is your typical protein, and this is 1,000 times more stable than that. But because that mutation changes the energy landscape, it leads to that amyloid fibril formation. And we can actually see the way that it changes the energy landscape with a technique called hydrogen exchange. So if you take a protein in water and put it in D2O, then the backbone amides on that protein will eventually come off, the backbone amide hydrogens will come off that protein, and they'll be replaced by deuterons from the solvent. So you can see that in a number of ways, but basically, if that backbone amide is making a hydrogen bond in the protein structure, then it's not going to exchange. So in order for it to exchange, that hydrogen bond has to break. There has to be some conformational fluctuation that exposes it to solvent, and that allows it to exchange. So if you look at the speeds that all these hydrogens exchange, then you can see basically how stable are all the different backbone hydrogen bonds in that protein. So this is wild type lysozyme. And really high bars mean that it exchanges very slowly. And you can see that the high bars go along with where the secondary structure elements are. So where there's no secondary structure, it exchanges really fast. So it exchanges very slowly because it's in all this secondary structure. This is the mutant right here. And you can see that this little part of the protein that's normally in this beta sheet still forms the beta sheet in the crystal structure, but it opens to solvent more quickly. That small part of the protein is fluctuating on and off more frequently than it would without that mutation. And it's actually this part of the protein that both makes initial contacts between monomers to start forming oligomers, and it gets integrated into the fibrils when this thing forms amyloid. So a small change in the energy landscape of one part of this protein ends up causing really significant medical consequences. And you can see just how that happens, like atomically, using this hydrogen exchange experiment. And this isn't just important for health, it's really important for designing therapeutics. So if you're going to design an antibody therapeutic that typically, like it's something that someone's going to inject into the heart, no, not into the heart, that injectable antibody therapeutic needs to be something that you can really concentrate down into a syringe, and it has to not form any kind of self-association, because for one thing, that will probably inhibit its function, and those like conglomerated antibodies can also be really immunogenic and is likely to be dangerous. So understanding protein energy landscapes is really important, not just in terms of the effect of mutations on disease, but also for engineering therapeutic proteins and for tons of other protein engineering applications. Okay, so the goal of this project is to accurately predict protein energy landscapes for any protein. And that's pretty ambitious because proteins can get really large and really complicated. But we're going to start with something very simple and then try to build our way up from there. So to understand why this is difficult right now, if you wanted to predict protein energy landscapes in a like machine learning type of way, let's look at the data that's available. So there's 150,000 structures in the PDB. And this is a great data set for machine learning on protein structure. It's one of the things that's driving like all neural nets to predict protein structure are trained on this data. It's really useful. Now let's say you want to predict protein stability. There's a database of 20,000 protein stability values, which is called the Proteome database. And that's not as great a database as the structural data, but it's still something. And my protease data isn't in there, but it's a similar source of large-scale stability data. Now let's say you want to try to predict protein energy landscapes. The only database that I'm aware of is the Start to Fold database that has energy landscapes for about 50 proteins. So right now, you really can't address this problem in a data-driven way. So how are we going to fix that? We're going to fix that first with a new high-throughput assay for protein energy landscapes. Hydrogen exchange-based assay that's going to show us not just is the protein stable or not, but precisely how stable is it and what are the energies of all the fluctuations that protein undergoes. We're going to start with very simple proteins and then build up in complexity. We're going to design many of the proteins that we want to study, because the proteins that we take from nature aren't necessarily the simplest, easiest to understand, best training set for a machine learning algorithm. It really helps if we can both look at proteins from nature and also design new ones and try predicting on those, design simple cases to test different aspects of energy landscapes. So I think being able to combine high throughput energy landscape measurement with designed proteins is going to be a really powerful combination. And then last, once we've collected all this data, then we're going to integrate that data into both machine learning models and physics-based force fields. So as kind of the first step in this project, I've started using the simple de novo designed proteins that I published a couple of years ago as the initial test set for this high throughput energy landscape measurement. So we're going to start with these types of proteins where, again, they're all about 40 residues long, but they have fairly different sequences even within a protein family. The sequences can be like 30% similar. Okay, so how do we do this measurement? We start with all the proteins that we want to experimentally test. And in this case, I'm right now doing this in batches of about 1,000. But one of the goals of this project is to scale that higher. And we can talk about how to do that. Once you have the 1,000 proteins that you want to test, You take the sequences of all those proteins, and you order them as an oligo library. So this is from Agilent or Twist. And then you take that oligo pool, clone it into a vector, and then transform that into a single culture of E. coli, and then, or cell-free or whatever, but I'm doing it so far in E. coli. And then this one culture will make all of these different proteins because it had this mixed pool of DNA. And then all these proteins have a his tag, so that can be purified away from the rest of the cellular material, so you just end up with the proteins that you want. And then we cleave off that his tag, so we're just left with that purified mixture of proteins. And then we take that sample, and that goes into hydrogen exchange LC-MS. So what that means is, if it's injected into a mass spec, there's a liquid chromatography step at the beginning. So each protein is kind of separated by liquid chromatography, and they elute at different times. But then you can also see which protein is which because they have different masses. And this is what you see for a single protein. So before that protein has ever gone into D2O, it has its normal mass distribution. And then we took that whole mixture, put it into D2O for 30 seconds, put it into a quench buffer, and then injected it into the mass spec. And then all the proteins got a little bit heavier because they picked up some of those deuterons. And then if this is 30 seconds, and this is a minute, and this is 5 minutes, and this is an hour, all the proteins just get heavier for each increased amount of exposure to the D2O, depending on how much of that protein is exposed, what are the energies of those fluctuations. And we see data like this for every single protein in the sample. So this is actually what it looks like if you just look at all the different proteins and when they elute. This is your LC injection. You start seeing proteins around 5 minutes. The whole injection is about 20 minutes long. Different proteins are separated out by mass, and each protein has a color here because the mass spec also has another dimension of separation that's called high mobility spectrometry. So there's basically 3 different dimensions that separate all these proteins from each other. And the reason that I'm showing this figure is that you can imagine like cramming a lot more spots into this and still getting like pretty well resolved data. So I don't yet know what the high limit of how many proteins we can measure at a time is, and that's going to be an important thing to push. So the largest library that I've tested so far is, I started with 1,300 DNA fragments, and I got really good data on about 730 of the proteins. So what happened to the others? Maybe they didn't express, maybe they don't fly very well in a mass spectrometer, maybe they were in a region that was so dense with proteins we couldn't deconvolute different signals, but still seeing 700 proteins at once starting from 1,300 is pretty good, and I want to go a lot higher. Okay, so how do we analyze this data? This is the part where I really lose people. So when I gave this talk a couple weeks ago, I made some new slides, and they thought the new slides were pretty good. So you can tell me if these new slides help explain what's going on. Okay, so this is a protein, and I've colored all the backbone positions according to basically what we predict, whether that position will open very slowly or whether that position is exposed to solvent and exchanges very fast. So red means hydrogen bonded and it exchanges slow, and the blue sites are kind of on the surface of the protein, and those sites exchange very fast. And to make it easier to think about this protein, I'm just going to talk about 3 sites. So we have a really fast exposed site here, a kind of medium exchanging site here, and a slow site here. And what that means is that at each time, there's some probability that this residue will exchange its hydrogen for a deuterium from the solvent. So at first, when it hasn't had very long, it's unlikely to exchange. But as we leave this protein in D2O for a really long time, eventually a stochastic event will happen that exchanges that proton for a deuteron. And because this is hydrogen bonded and buried in the protein, it's very slow. For this kind of medium exchanging site, because this part of the protein is opening up more often, this will eventually go fully exchange, like every copy of this protein will fully exchange that proton sooner than for the red site. Please.
Sorry, I'm not sure if you mentioned it, but how do you make these individual predictions? I'm not actually trying to predict anything about these proteins. I'm just saying, imagine this is the way that this protein is, then this is what the data would look like. So
please just arbitrarily place fast, medium, and slow on a de novo protein?
I'm using a somewhat realistic example. So basically, I'm trying to explain how do we go from our data to understanding the protein. And right now, I'm saying, imagine the protein were like this. This is what the data would look like. Okay. So for this particular site, it goes to fully exchanged faster because that part is opening up more often than the red site. And this is a very exposed site. The blue site is a very exposed site that basically fully exchanges very quickly. So the way our experiment works is we take time points for this whole mixture, we put it in D2O for a set amount of time, and then we inject it. We have another sample that went for a longer time, and we inject that. And so if we have a very early sample, then there's many copies of this molecule in the mixture, and so we see a distribution of how much mass it's picked up. So for every copy of this molecule, the blue site is already exchanged at this time point. So we have at least one added mass unit. The orange site has exchanged a little bit, and the red site has exchanged a little bit. So sometimes we see 2 added mass units, but we very rarely see 3 added mass units. So this is what our mass spec is telling us when we look at this protein at this time point. And then at a later time point, now the orange and blue protons have fully exchanged, and the red proton has exchanged about half the time. So we see at least 2 exchanges for this molecule, and we see some amount of 3 exchange when this red one is also gone. And then if we look at a really late time point, then all 3 of these sites have pretty much exchanged. So almost every copy of this molecule has had 3 exchanges, but for some of them it's only 2. Okay, but I'll get your question in 1 second. Well, yeah, sure. What was your question? Did you have a question?
So the fast one should be relatively identified, but between the medium and slow, I mean, you imagine there's like, it's not just 3 categories, it's a...
Right. So what we're really seeing is one different curve for every site on this molecule. So every different amide is exchanging at some rate that's unique to it. And then so we see this wide distribution where at an early site, this is what we see by the mass. At an early time point, this is what we see by the mass spec. And at a later time point, there's been more exchange. And then at a really late time point, this thing has exchanged basically as much as it can. And then the way we're analyzing the data is fitting these curves at every time point to basically extract the set of rate constants behind these curves. So I don't know which site is fast and which site is slow, but I know the distribution of how many fast sites, how many slow sites there are. So we get like 40 of these curves, 40 of these distributions, because we measure this at 40 different time points. And then that's going to give what I'm calling the free energy spectrum, which is just all these rates. Well, they're opening energies. How often does that site open? Organized from the most stable site in the protein, and then a kind of medium stable site, and then these are the really unstable sites. So this is what our data at the end of the day looks like for each protein. It says how many residues are there that are really stable, how many residues are there that are medium stable, or down this whole line to the number of residues that exchange very, very quickly. So we get this data, we use this data to infer these curves, and then I'm going to draw the data like this, which is the distribution of opening energies in each protein. Please.
Just experimentally, so this is all intact mass.
Yeah.
Okay, so there's no tryptophanization and there's no MS-MS.
That's right.
Okay, so you aren't able to, and then so how do you identify each protein as unique? Do they all have unique enough masses?
Yeah.
And you can see that?
So before I do this experiment, we just run an undeutorated sample, and then there we can do MS-MS, identify all the proteins, and then in every run of the deuterated samples, every protein elutes at the same time and has the exact same ion mobility drift time. So the protein appears in the same place, and then we can just watch its mass increase. But we only do the MS-MS the very first time on the undeutorated sample. Any more questions before I go on?
Why not do the SMS on the deuterated sample?
Because first of all, when the masses of the peptides have changed, you don't know what peptide came from what protein. But we actually are going to kind of start doing MS-MS, but that's a longer discussion. But so I should say a long-term goal of this project is to not just restrict ourselves to getting this kind of overall protein data, but what I'm going to show you for the rest of the talk, and like we want to get actual experimental site resolved data in a high throughput way. But what I'm going to show you for the rest of the talk is that we can learn a lot even just from this overall protein data. Okay, so what can we learn from the full energy landscapes of 1,000 proteins? Well, one of the first things that we can learn is that even two proteins that have basically the same fold can have really different energy landscapes. So these are two proteins that both fold like this. They both have this hairpin and then helix and then hairpin fold. So a 4-stranded beta sheet made out of 2 hairpins and a helix. This is like the protein G fold, except it's only 40 residues long. And these are 2 different proteins that both fold like this. And these 2 proteins are really interesting. I just picked 2 out of the set of about 400 of this fold that we've looked at. These proteins are really interesting because the blue protein is more stable than the red protein in terms of the most stable chunk. The most stable chunk of the blue protein is more stable than the red protein. But then after about 17 residues, it really falls off, and then some parts are fluctuating much more than the red protein. So even though the red protein is less stable, it kind of keeps itself all together more than the blue protein does. It has some chunk that's fluctuating off all the time. And in order to get positional data about these 2 proteins, we actually looked at them by NMR, where you can see the exchange rates at each site. And for the red protein, the flat one, it looks about as you'd expect. There's kind of similar opening energies along the sheet, around 2.5 kcal per mole, 3 kcal per mole, and then the helix is the most stable part. For the blue protein that has that has the fluctuating part, the first hairpin is relatively stable, and then the helix is very stable. But this second hairpin is coming on and off so frequently that we couldn't even see any protection by NMR. So these proteins have the same fold, and I'll show you that again later. These proteins have the same fold. This is actually a more stable protein. Like if you did a quantity belt or some other method that you're used to look at protein stability, this is the more stable one. But it has this really dynamic part that's always coming on and off. And with data sets like this, I like to understand why does the sequence of these 2 cause these different energy landscapes. Let me show you one other example. These are 2 different proteins that have a 3 helical fold. And again, the blue protein is more stable than the red protein in terms of the most stable chunk. But the red protein is more cooperative. For the less stable parts, it keeps itself together more of the time. But some chunk of the blue protein is really fluctuating off a lot. Again, we did the NMR and looked at the site resolved dynamics in these 2 proteins. There's 3 helices, helix 1, helix 2, helix 3. In helix 1, the blue protein is more stable. In helix 2, the blue protein is again more stable. But in helix 3, we couldn't actually measure any protection of those residues because it's fluctuating so much. But the red protein has about the same energy as the other 2 helices. These are the design structures of these 2 different proteins, and you can see that they were designed to fold basically the same way. And in general, the proteins really do fold pretty much exactly as they're designed, and I'll show you that again in a second. These 2 proteins were designed to have helix 1 in a slightly different place, but we see really different energetics in helix 3, where helix 3 for one of them is fluctuating on and off all the time. So you might ask, for these proteins where one part doesn't seem that stable, do they actually, has the protein changed from how it was designed and it doesn't really fold correctly? So here I'm showing you the 2 different non-cooperative proteins that we just showed, the very dynamic ones. And we actually solved full NMR solution structures of these 2 proteins. So in white is the design model, and the bundle of rainbow is the NMR solution ensemble. And this mixed alpha beta protein folds exactly as designed. The 3 helical one folds almost exactly as designed. It's a little bit off. But anyway, you can see that 99% of the time it's folded as designed, which is what the NMR solution ensemble, which is what the NOEs are saying. But 1% of the time, or some very interesting amount of the time, this whole hairpin is fluctuating on and off this molecule, and this red helix back here is fluctuating on and off this molecule. And we never would have been able to see this with an assay like protease stability. Both of these proteins are much more protease resistant than an unfolded sequence, but they have this really interesting dynamics that's only captured when we go to this higher resolution method that we can now do in a high throughput way. Okay, so the last question is, how do sequence and structure determine protein energy landscapes? Why are these proteins different from each other? And like, this is going to be a great question to answer when we have 20,000 data points on this, but I'm going to show you kind of the very first steps that I've taken in a really simple way, just using this data set of about 1,000 proteins. Actually, I'm just going to, from now on, I'm just going to look at the 400 proteins that I've measured that have this fold. So we're going to look at a pretty small set. Okay, how do sequence and structure determine protein energy landscapes? What we see first is that these energy landscapes basically differ from each other in two main ways. And you can see that with a method called principal component analysis that some of you are probably familiar with. It's a way of taking high dimensional data and looking at correlations within those dimensions to collapse it into some lower dimensional space. So if you take all these energy landscapes and do PCA on them, then you get basically two main principal components that capture almost all of the variation in these energy landscapes. The first principal component is how high is the landscape. So on one end, there's just really unstable proteins like this blue protein, and on the other end, there's really stable proteins like this pink protein. The second principal component is describing the shapes of these energy landscapes. So you can have a very flat energy landscape, like this blue protein, where the whole protein basically opens up at more or less the same energy. There's not like parts that fluctuate very frequently when the rest of the protein isn't opening. Or a very non-cooperative protein, like this red protein, where there's a really stable chunk, but then this chunk down here is fluctuating on and off very frequently, especially compared to the other proteins in the same fold. So we can basically describe all these energy landscapes using these two different dimensions. How high is the whole landscape, and what's the shape, cooperative or non-cooperative? And here I've plotted the 400 different proteins in this fold in this new coordinate system, where there's really unstable proteins over here, really stable proteins over here. These are very cooperative proteins with flat landscapes, so nothing's fluctuating. And these are proteins where some piece of that protein is really less stable than you expect based on the rest of the protein. So now the question is, why are the different proteins where they are? Why is one sequence up here and another sequence up here? And for this little toy example, I only have 400 data points to play with, and I haven't tried looking at it in too much depth, but I'll show you a little bit of what we found. So we can quantify a lot of the features of these proteins, like how hydrophobic is it? How many prolines are there? What does Rosetta calculate the energy to be? And I tried taking 6 of those features that we know are pretty important, and well, I tried taking a lot of them and building different linear regression models to basically predict, using a linear regression on a few features of these proteins to predict where is it in this dimension and where is it in that dimension. And so the models that I'm going to show you use only 6 features of these proteins. So this is using this 6-term linear model to predict principal component 1, and you can see we capture about 1/4 of the variation in the data using just 6 features. And again, for principal component 2, we get about the same correlation. And so the models that I'm showing you here are not cross-validated, but if you do this in a cross-validated way, you end up with about the same correlation. So you can capture about 1/4 of the variation in both overall stability and cooperativity with just 6 simple features. And the reason why I think this is interesting is that different features of these proteins affect their energy landscapes in different ways. So these are the coefficients of those 6 features in these 2 different models. So this is the coefficient in the linear regression of principal component 1, and this is the coefficient in the linear regression of principal component 2. And you can see that something like the number of prolines in each protein has a big effect on how cooperative that protein is, whether it's all or nothing or whether there's part that fluctuates a lot. But it's really not doing very much for the overall stability of that whole protein. But something like counting the number of hydrophobic residues, that's having a big impact on the overall stability without really affecting the cooperativity that much. So different features affect these energy landscapes in different ways. And again, this is just the simplest thing that we could do, like a linear regression with a very small number of features on a relatively small data set of only 400 proteins. But you can imagine how this can develop when we have tens of thousands of these measurements and apply nonlinear models, apply more complicated models to hopefully ultimately be able to predict from sequence and structure what are the energy landscapes of these proteins. So the long-term goal of this project is, can we build new machine learning and physics-based models that are quantitatively predictive of these energy landscapes? And so I'm just going to list quick bullet points on a lot of the different future directions. We're going to collect a lot more data very efficiently. We have a great mass spec for doing hydrogen exchange with a robot attached to it that basically does all the mixing. It takes a sample, puts it in for 10 seconds, has the 30-minute sample running so that one gets injected when the 10-second one's finished. It's sitting in boxes right now, but all that should be set up. All that should be set up in the next like 2 months, I hope. But it's going to be really great for collecting this data really efficiently. We're going to start moving up to larger proteins. There's nothing about like all the DNA methods and all the hydrogen exchange methods are perfectly applicable up to whatever size of protein. So we're not limited to working with these small proteins. That's just a good starting point. We're going to try to get local information about what are the stable and unstable parts. And there's a lot of cool ways to do that that are going to be really exciting for this project. We want to look at not just designed proteins, but also natural proteins and their disease mutants. When I showed you the linear regressions, those were just linear regressions on based on properties from a single structure of the protein. But you can imagine maybe if we do some type of conformational sampling, whether that's conformational sampling in Rosetta or coarse grain modeling or full atom electrodynamic simulations, whether data from that type of conformational sampling would be useful for predicting these energy landscapes. We're going to use this data to optimize physics-based force fields and push the machine learning beyond like the simple linear regression that I showed you. So thanks everyone for your attention and really great questions. I want to just acknowledge a few people. I started this project at the tail end of my postdoc in the Baker Lab. Mike Gutman, another lab at UW, taught me how to do all the hydrogen exchange. And he's a really great resource. I just came back from a hydrogen exchange conference in Banff, where like a quarter of all the posters had Mike's name on them. So that was really, not because they were from his lab, but just because he helps out so many people in the field. So that was really nice. And all the NMR data that I showed you came from the Aerosmith lab in Toronto. So thanks for your attention. I'd love for people to come work on this project, and I'd love to have more discussion if you have more questions. Thanks.
We have time for a few questions.
So you always, when you were looking at those unfolding landscapes or the hydrogen exchange, yeah, things like this, you always refer to, when you see a shelf like that, you refer to it as a chunk. To what degree do you know that that's actually, or how often is it the case that that's like a chunk in primary sequence as opposed to like a number of residues that just happen to have the same?
Yeah, that's a great question that we do not know the answer to. So that's one of the reasons why it's really important to move this from just looking at the overall protein level to getting some more local resolution about what the stable and unstable parts are. I can tell you that in the two proteins that we followed up on, these two, it was definitely one contiguous region that was much less stable than you expect. Here it was, this whole hairpin was kind of an unstable part. And here you can see like helix 1 is normal, helix 1 is normal, helix 2 is normal, and helix 3 is really unstable. So that's only two proteins though. I don't know if that'll continue to be. Actually, I've seen, there's a third protein that I didn't show that's again also one continuous region. But I don't necessarily expect that to hold up across thousands of proteins. So it's a really important goal to answer that question. Back there?
Do you expect to see any types of proteins interacting with each other in the solution, or can you
Yeah.
after that?
Yeah, that's a great question. It's a little bit of a concern. It's not a huge concern. So first of all, it could totally happen. The concentration that I inject into the mass spec, well, the concentration, so the protein sample is about a mg per mil, but then when we put it into the D2O, it's only a tenth of a mg per mil. So it's a fairly dilute protein sample at the time that it's exchanging. Each individual protein is at a really low concentration because each individual protein is only a thousandth of a tenth of a mg per mil solution. So if there's going to be some specific interaction, it would have to be a really strong interaction. There could be some generic, non-specific interactions that might happen. I will say that we followed up 9 of these proteins with this site-specific NMR data, and the NMR data, which is on just a sample that has only 1 protein in it, is really consistent with what we see by the mass spec hydrogen exchange on the big mixture. So at least it doesn't seem like we're seeing problems, seeing weird effects of having a mixture of different proteins. But something that could totally happen that we need to look out for.
I don't know what size range you're thinking, but as you move larger and larger, you may not be able to resolve the structure using NMR. Do you, like, what's that like a bit of structures with NMR? And if you run to that limit, how do you think you can adapt?
Yeah, so one nice thing about using the design proteins, or any protein that we want, we need to have, it helps to have a pretty good idea of that structure. And with design proteins, we can be really confident that they actually fold as designed because of all the other controls that we've done in the past. And that's held up even for these non-cooperative proteins. You're right that sometimes if we see a protein that has a really weird energy landscape, then we probably want to get some kind of higher resolution structure. NMR structure determination, I don't actually know where it tops out. Might be like 150 residues or something. Then we could try to crystallize proteins. There is actually a normal hydrogen exchange technique for getting really detailed site-resolved information on one protein at a time. So we can use hydrogen exchange to get, even if we can't do NMR, to get a really detailed picture of what the protected and unprotected parts of each protein are. Does that answer your question? Sure.
Yeah, just want to follow up on Isaac's question. So what are the prospects for looking at how the individual parts of the protein are exposed or not? So, I mean, you have MS-MS, but HTX maybe exchanges during MS-MS, I don't know. And then trypsinization takes time, which can decrease the resolution on the D2O exchange. So I wonder how that's going to plan.
Yeah, so the classical way to do it is you do the exchange normally on the full proteins, and then when they get injected into the mass spec under a quenched condition where nothing's exchanging, the first thing that happens is they go over a pepsin column, and that cuts up each thing into peptides, and then you see how deuterated each peptide is. That's going to make your sample a lot more complicated because now you have like 50 peptides that came from 1,000 proteins, so it's a lot more complicated to sample than just 1,000 proteins that you started with. But maybe you can do it on 100 proteins. Maybe we can take our set of proteins. We can take our set of proteins like this, and we say, well, everything down here is really cooperative. It's going to have the same energies all across the protein, so those aren't interesting. Everything up here has some really interesting instabilities where some part is really unstable. Then we make a smaller sample that's this, and that's the sample that we put over the pepsin column so that once everything gets broken up into peptides, it's still controllable. There's also a different MS-MS technique called electron transfer dissociation or electron capture, ETD or ECD, that can fragment the protein as it's eluting and in a way that doesn't scramble where the deuteriums are and lets you actually select what you're going to fragment. Same as HCD. But that can also be really powerful, and we're going to explore that like as soon as the mass specs run. Please.
So how long is one HTX experiment like this?
Each time point is about a half hour. Like the injection, so you can incubate the protein for however long, like 10 seconds to 24 hours. But then from the time that we inject to running the LC to washing the machine to when we can inject the next sample, that's about a half hour.
Okay. So yeah. So I was just thinking it would be very interesting to test like small molecules on these libraries to like actually see like what changes the energy landscape of binding the dynamic, because you get dynamic resolution like
Yeah, which proteins respond to TMAO, like a protein stabilizer? Which proteins are more affected? Are they all affected equally by urea and guanidine? Which ones are affected more or less? Which ones are affected more by ionic strength? Maybe some of these proteins are stabilized more by electrostatics, so then when we go to a high salt condition, some of them lose a lot of stability, whereas others don't. And then we can start quantifying all these things by doing the entire sample under the same condition in a really controlled way.
So is that feasible to do to screen a lot of molecules or something?
Well, I mean, you couldn't screen like a million different compounds for binding, but we could. But like, if you say so, each time point's a half hour. We can do 40 time points in a day, and that's about all you need for one of these mixtures. So you could do 10 different conditions on that full mixture in 10 days or something like that.
Yeah, that's cool.
Let's thank Dr. Rocklin.
Thank you so much.